The presentation today will be painfully technical, for which I apologize now. Therefore, I will not take offense if you depart during the presentation. I simply know of no other way to expose the hypocrisy of the conjectures based upon the evidence. Nanocar syntheses will be considered as nanosystems comprised of molecules working in concert. I will use nanocars to show you how molecules come together, and then we'll move to the biological system. This is not a philosophical talk, it is a technical talk. We're not considered or scientifically unknown entities that have been proposed to have seeded life on Earth, such as a design agent, personal or impersonal, or panspermia. Neither of those will be considered. The latter one being that some aliens from another planet seeded life here. That just begs the question. It offshoots origin of life to another planet. An opinion will be rendered showing that the strongest evidence against the proposals of current prebiotic research is the researchers' own data. The data seem to be directing researchers down paths of futility. This is a simple life form. It is a cell. So it is what is considered a simple life form. A cell is a factory, an absolute factory. You have tubules, for example, that move materials from point A to point B. When you go into a factory, what happens? You look overhead and materials are moving. Here you have tubules that form to allow you to transport things, and then the tubules break up and form somewhere else. Why don't they just leave the tubules? Because the cell would become too rigid and you'd run out of the tube material. So it deconstructs and reconstructs. There are entities in here and organelles which their function is absolutely extraordinary. This isn't a simple life form, just one cell. Abiogenesis is the prebiotic process wherein life, such as a cell, arises from non-living, simple organic compounds. On our planet, this is what it is. In our universe, this is what it is, as far as we can tell. We're the only ones here so far. But certainly on our planet, it's carbohydrates, nucleic acids, lipids, and proteins. Proteins are polymers of amino acids. Carbohydrates are sugars. So it's just these four classes of compounds, carbohydrates, nucleic acids, lipids, and proteins, make up the substance of life as we know it. All of this is needed long before evolution can ever begin. This predates evolution. This is long before evolution. This, you have to have the first seeded life. You have to have the first cell before you can start talking about evolutionary processes. This predates all of that. Collective cluelessness. We have no idea how the molecules that compose living systems could have been devised such that they would work in concert to fulfill biology's functions. We have no idea how the basic set of molecules, carbohydrates, nucleic acids, lipids, and proteins, were made and how they could have coupled into the proper sequences and then transformed into the ordered assemblies until there was the construction of a complex biological system, like a cell, and eventually to that first cell. Nobody has any idea on how this was done when using our commonly understood mechanisms of chemical science. Those that say that they understand are generally wholly uninformed regarding chemical synthesis. Those that say, oh, this is well worked out, they know nothing about chemical synthesis. Nothing. Further cluelessness. From a synthetic chemical perspective, neither I nor any of my colleagues can fathom a prebiotic molecular route to construction of a complex system. We cannot figure out the prebiotic routes to the basic building blocks of life, carbohydrates, nucleic acids, lipids, and proteins. Chemists are collectively bewildered. Hence, I say that no chemist understands prebiotic synthesis of the requisite building blocks, let alone their assembly into a complex system. That's how clueless we are. I've asked all of my colleagues, National Academy members, Nobel Prize winners. I sit with them in offices. Nobody understands this. So if your professors say it's all worked out, your teachers say it's all worked out, they don't know what they're talking about. It is not worked out. You cannot just refer this to somebody else. They don't know what they're talking about. Synthetic chemistry and nature's synthesis. To the synthetic chemist, making molecules is one of life's greatest joys, affording a sense of accomplishment and prize when a newly devised route has yielded the long-sought predicted product. Yet humankind's complex synthesis is child's play in relation to nature's sophistication, which has exquisite precision, often nearly 100% yields, with pre-designed correction or scrubber molecules that are employed when the yields are not 100%. The scrubber molecules go and they take the bad ones out. Nature's systems perform bottom-up construction at phenomenal rates. So it's bottom-up, and we'll talk about that in more detail. At phenomenal rates, the rate at which DNA is synthesized is just utterly, utterly amazing. All leading to full-sized functional systems that are plentiful on Earth. They're ubiquitous. This is seen in what some might call a simple cell, up to a fully functional human being, or elephant, or blue whale. Every breath we take and all that we see, touch, taste, smell is made of molecules. Everything. That's it. It's all based of molecules. Our memories, which capture joys, pains, or emotional struggles, are embedded in molecular structures and molecular potentials. When I'm talking to people, I often get sidetracked because I'm looking in their eyes as they're speaking, and I'm thinking of the rhodopsin, of the changes in the cis-trans isomerization that's occurring as the photons are hitting their eyes. And as I look in their eyes, I'm thinking of their brain, of the actual protein synthesis. So it's electrons as I'm speaking to you. It's electrical potentials that are forming in your brain that are giving you these thoughts as I'm speaking. And then those go rapidly into protein synthesis that give you these short-term memories. And then tonight, as you're sleeping, that protein synthesis starts turning into hardwired connects in our brains that give us our long-term memory. So if I hit the table, you don't forget that I hit the table because it's already gone into protein synthesis. And that becomes hardwired interconnects, so that tonight as you're sleeping, it starts going into hardwired interconnects where you have molecular structure change. And so that you won't forget that I hit the table that night when I was there at Waterloo. This is what happens. This is how every remembrance occurs through molecular structure changes. It's all based in the molecules. Absolutely everything is molecular based. Chemists study molecules. Synthetic chemists make molecules. Synthetic chemists design the routes to the plethora of pharmaceuticals, dyes, perfumes, and materials that make our lives healthier, more productive, more enjoyable. For example, the one thing that has saved more lives by far than anything else in the world is sodium hypochlorite, the simple molecules NaOCl, used in the detoxification of water. This simple compound made from chlorine and sodium hydroxide saves millions of lives every year in the developed world. If it were used in the underdeveloped world more often, it would save millions of lives there because more people die of waterborne diseases than anything else on Earth. Target selection. How do we choose the target molecule for a specific use? How does a chemist choose this? How do we know a priori the behavior of the target? In some cases, we've seen it work in a certain way in related systems or molecules like it, responding in the way that we wish so that we target that and other molecules with similar structure. In other cases, we do not know for sure, but we conjecture that it could have certain desirable properties based upon its molecular weight, its shape, its addends or functionalities that would render it as acidic as we would like, or as water-soluble as we wish, or having the shape to fit in an enzymatic cleft. That's how we choose the target. Choosing a target is hard. It takes years of training and forethought to be able to select the correct target or class of molecules for a specific function. And with our best efforts at prediction based upon experience, the literature, and computer modeling, it often takes numerous attempts to choose a target suitable for a desired task. But if one had never seen a complex analog, to what do we target? So remember nature's problem here. You want to make a functional system. You don't know what to build to make it. You've never seen the target before. So nature's problem is much bigger than our problem when you consider a prebiotic world, before biology world. Target selection is often based on analogs. It is intellectually baffling as to how nature could have progressed toward complex functional systems over and over again from basic molecular structures when no basis for the collective function had pre-existed. And in nature, this has to be done correctly over and over again. The failed attempts are not innocuous. They deplete precious starting materials from the previous steps, and those starting materials are not easily resynthesized because the conditions for their previous steps are now expunged. They no longer exist. It wasn't written down in the chemical literature. And the non-functional products remain in the system, contaminating it, gumming up the works, so that further desired reactions are inhibited. That is, unless there resides chemical scrubbers that can selectively remove the unwanted compounds while being sophisticated enough to not degrade the desired compound. But how does a prebiotic system know a priori what is desired if it cannot assess its ultimate functional activity until several steps forward? So one needs a separate set of scrubbers for each product step. And those scrubbers or correction molecules are themselves highly sophisticated in structure and activity, so that each of those correction molecules needs to be designed specifically for the task. But in a prebiotic world, who designed it? There's no designer. Remember, we're not considering a designer here. We're not considering somebody seeding this. These are just the beginnings of the befuddlements when chemists consider a prebiotic system. How is synthesis performed? This is how we as synthetic chemists perform synthesis. You start with a target, and you work your way back to conveniently available materials that in a simple step could yield that target. And then you work your way back from each of these intermediates into sub-targets, and you work your way back and back and back until you hit readily available starting material. And then you start pruning this tree. Why do synthetic chemists use retrosynthetic approaches to build complex molecules? Because without the retrosynthetic approach, discerning one's way to the desired products is far too complex, leading to dead ends that are overwhelmingly abundant, generating massive amounts of undesired products and exhausting precious supplies of starting materials that might take huge amounts of efforts to prepare. But nature can't perform retrosynthetic analyses if we presuppose that the starting points progress to a non-predefined endpoint. And again, this is utterly perplexing for the synthetic chemist. How could this have happened in prebiotic chemistry? How do you go from a starting material to a product that's a complex product? What we do is we take the product and we work our way back slowly. But nature doesn't know what its product's going to be at the end. Doesn't know. Just blindly going along. The chemist or team of chemists then goes to the laboratory trying a host of reaction types and permutations to proceed along the synthetic course, often optimizing at each phase en route to the desired intermediates and then to the final product. Each step is considered with chemical mechanisms. How many people in here are synthetic organic chemists? Yes. I've got one. Anybody else? All right. You have to confirm for me. If I tell a lie, just say, liar, liar, pants on fire. Okay? Each step is considered with chemical mechanisms, the precise steps in the specific reaction site modifications, while weighing the possible different reaction rates for change at each of those potential sites to accomplish the task at hand. Purification then ensues, since few reactions ever afford 100% yield with no deleterious byproducts. Purification is often a painful process, trying to separate the good from the bad, the desired from the undesired, when the undesired is often just ever so subtly different in structure than the desired product. We spend lots of time purifying. The differences can be as minute as to be the mirror image of what we want, making separations a time-consuming and challenging task because one often needs complementary mirror image pure structures to purify the desired products in mirror image purity. Interestingly, many of nature's molecules that she uses for biological systems are mirror image pure, or what we would call homochiral. If the byproducts are left in the reaction, those impurities result in complex mixtures that become so overburdening that they render future reactions almost impossible to properly execute. If you don't purify, you go through a few steps without purification, you've got so much junk in there that all that junk consumes your starting material and you can't get product out of it anymore. How does nature purify? Well, in biological systems, number one, it uses very, very high yields, not nearly 100%, not all the time, but most of the time. And then it has chemical scrubbers that get rid of the bad ones. And when things run amok, we get cancer, where the scrubbers didn't scrub out what they were supposed to, or pull out the base pair that was causing the problem in the DNA. But how do you do this in the prebiotic world? Before there was chemical scrubbers designed. The chemical scrubbers are themselves at least or more sophisticated than the molecules that you're trying to make. Structure identification and process rates. And then the laborious structure determination process takes place after each synthetic step in purification, using a host of different spectroscopic, spectrometric methods to confirm molecular structure. That's the way we as chemists do this. If we've made the wrong molecular intermediate, it will throw us off all subsequent steps. If we've made the wrong thing, it's going to really mess us up. So we'd better get the assignment of structure made correctly. And often the intermediates to the products can be unstable in air, sunlight, or even room light, or in water, or sometimes we need to work fast, in seconds or minutes, for example, and under strict exclusion of those conditions that might affect it. Abiotic systems do not have these tools or readily accessible strict controls. Remember, this is happening in some cave somewhere, in a pool. It doesn't have strict controls. Much harder for nature. Assembling molecules to build a functional nanosystem. I will show you the building of a nanosystem. It bothers people when I say that there's few people on Earth that know as much as I do about building molecules into a complex system. And the reason I say this is that there are only a handful of us that build molecules where we would design complex functionality that we're going to build from this, like a nanocar, where we're going to take molecules and build a functional nanosystem. There's only a small number of us in the world. So I know at least as much as any one of that small number. So I certainly think I know more than most high school students, than most college students, and generally I know more, I hope, than most of the people that I teach in my lecture. Building a nanosystem is not easy. Let me show you the steps that are involved, and then we'll move into the biological constructs. Why a nanomachine? Why do we want to build nanomachines? Well, why Mona Lisa? What's her function? What's her application? It's just aesthetically beautiful. Even if we had no application, it'd just be intellectually interesting in academia to do this. Say you have a tree. What do? You cut down that tree, you go ahead and you make a table. That is top-down construction. That is the way we build. We want to build a podium, find a big tree, cut it down, build our podium. Nature goes the other way. It goes from the bottom up. These seeds go to the oak tree. Then from that, you go ahead and you build your table. Well, you know, it might be that not too long from now, people will learn how to go from the seeds to the table. Just modify the DNA. Why bother go through into the tree? Just go from the seeds to the table. I mean, you laugh. This is not that far-fetched on things that people consider. Or what we're considering is, could we just take atoms and carbohydrates? Carbohydrates are what make up the tree, and just go directly with nanomachines and build a table. So it's bottom-up construction directly to the table. So that in 100 or 200 years, we'll build skyscrapers this way. You laugh, but how was the tree made? How were you made? You were made from the bottom up, from smaller molecules. They came together, not some of that by thermodynamic interaction, meaning that they lined up and you begin to get assembly, like molecules line up in a self-assembled layer, A, or A, B. But you cannot get complex function by having regular order. Regular patterns of order, A, B, or A, will never give you complex function. We know this from computer science. You want complex function, you have to have non-regular assembly. And this is what happens. So how does that happen thermodynamically? Well, what happens is you get these molecules called enzymes, which are nature's nanomachines, and they start building up. That's why you eat a bagel this morning, and by this afternoon it's a part of your ear. How did that happen? It's because enzymes break down those carbohydrates, they break down those molecules, and they reapply them and reattach them where a cell needs them. This is what enzymes do. They do this all the time. It is happening in your bodies right now. Thousands of enzymes working and doing construction in your body. This is how things are built in nature from the bottom up. What we want to do is we want to build buildings this way. And we are laying the groundwork for this. So in a few hundred years, people just bring in raw materials. The thing builds. Sounds like science fiction. There's some bamboo, for example, can grow 3 feet tall in a single day. So biology actually can build very, very fast. And the sophistication in that bamboo is more than the technological sophistication in this room. By far at the molecular level. Amazing sophistication. It's not magical. It's not magic. There are reasons why these can be built this way. And what we want to do is mimic this in an abiological system, in a non-living system. So these are nanocars. And they're 2 nanometers by 3 nanometers. We can part 30 to 50,000 of them, depending on which size nanocar we use, across the diameter of a human hair. So they are really, really small. And you can see pictures of them. You see the 4 wheels because in STM you see the highest density of state part of the structure. Here's a nanometer. Because it's 2 nanometers by 3 nanometers, it's a rectangle, and so we know its direction. It's facing this way. It's going to turn and move out across this surface, and there it goes, moving across this surface. This is a gold surface, and they collided right there. That was the first nanocar collision ever recorded. These are 1 atomic step high islands on the gold. So we built these cars intentionally so that whether they started going forward or back, it didn't matter. However they landed, it didn't matter. Top and bottom was the same. This was designed in there. This was our 4th generation car. Fully rotating axles. And we know that these are indeed not sliding on the surface, first of all by the directionality of how they roll, but then when we make the trimer, these only pivot on axis. These don't roll. If they were just sliding like a car on ice, the trimer should slide as well. It doesn't. It just spins on axis. We built this so that the axle had 360 degrees rotation relative to the chassis. When my 2 sons were young, you get them these cars and they just drive them all day, you know, and the axle can spin around relative to the chassis. That allows that car to flip over, it allows it to go up over steps, and we built this functionality. Why did we build this? Because we could never get up these 1 atomic step high islands, so we built in suspension. We thought about this, and then we built it to do this. And that allowed us here, you see this 1 atomic step, this is 1 atom higher in gold than the background surface. And then there's a nanocar, drives right up it, and then comes back down the other side. These are just screenshots. One comes up, and they just go right over this. With these monster wheels, they go right over it. They go right over it, single atom high. I mean, it's amazing what they can climb. We built the functionality into it. It doesn't change on its own. Doesn't change on its own. You build the functionality into it. You can put a tip, an STM tip in front of the car and it'll follow the tip because of an electric field. This is how we're going to control these in years to come. We'll use electric fields. And you just bring in enough cars. You say, how much cargo can a car carry? It's not how much cargo can it carry. It's how many cargo carriers have you? We make these 10 to the 23rd at a time. That is a huge, huge number. Avogadro's number, 6 times 10 to the 23rd, is huge. How huge is it? When you take a swallow of water, that's 18 milliliters of water. That is 1 mole of water. 6 times 10 to the 23rd molecules. 1 swallow. If you had 6 times 10 to the 23rd sheets of paper, and you just stacked up a sheet of paper, if you have 500 sheets, it's about that high. If you had 6 times 10 to the 23rd sheets of paper, that stack of paper would reach from the Earth to the Sun 400 million times. So you'd have 400 million stacks of paper from the Earth to the Sun if you had 6 times 10 to the 23rd sheets of paper, which is exactly how many water molecules you have in one swallow of water. Do you see how small molecules are? Now, if you can use these as cargo carriers, if you could encode in water, in molecules the size of water, information, you could put all the information of humankind in this much water. All the information that's been in everybody's brain for the last 5,000 years of human history. All of it. And we keep doubling information. Every 2 years, we double all the information that we've acquired over the last 5,000 years. That's how fast our information is growing. Now, accessing it, that's another problem. But that shows you how small these are. You put the tip on the side of the car, it can't move sideways. Then many people said you don't have a car until you have a motor. So we built these motors. These motors only rotate in one direction, so they only rotate unidirectionally. The motors were designed by a man named Feringa in the Netherlands. And then we took his motors and we built them into the nanocars such that it would be at mid-chassis, and you photolize this, and the motor starts spinning around. And the reason that these spin unidirectionally is what happens when you shine light on this twisted double bond. There are two elements of chirality here. There's a twisted double bond, and there's a stereogenic center right there. So what happens when you shine a light, that twisted double bond goes orthogonal. And it can relax back down either way. But because there's a neighboring stereogenic center, the two directions are diastereotopic, so they're necessarily different in energy. And so it goes down the lower energy side. And then it undergoes this helix inversion, and then continues on around. And so the idea is to build this in at mid-chassis, and we did it. So we built up molecules, and it's a much longer synthesis than what I'm showing here. I'm just showing you the last basic components of how we make wheels. And now you see these wheels are no longer fullerene wheels. We had to switch to carburein wheels. And the reason we had to switch to carburein wheels is because we first made it with the fullerene wheels. And then when we'd shine light on it to activate the motor wouldn't turn, because it would transfer its energy to the fullerene wheels. We didn't know it until after we built it. You think nature didn't get stuck in holes like this? You think it knew exactly what to build? Didn't even know what it was targeting, and at least we knew we were going after a nanocar. So lots of synthetic roots give you things that you never expected. And so we had to switch to carburein wheels so that we could activate the motor without it transferring its energy to the carburein. How did we know carburein? Because it doesn't have a pi system. Nature never knows stuff like that. Certainly in an abiological system. But we knew that, so we had to change wheels. Then it turned out that that motor worked, but it only spun at 1.8 revolutions per hour. Slow motor. But then we found that if we took out the sulfur atom here and closed that up into a 5-membered ring, then it would spin at 3 million rotations per second. Now we're talking. 3 megahertz rotation. Small change gives a huge difference. We had literature precedent that told us this. That means we go to books, to journals, and we read this. And there was precedent that this could happen, and it happened that way. You don't just know this psychically. Nature doesn't just know these things. It has to try lots and lots of combinations. How do you pull a sulfur atom out of here and go to here? We'll get to that. Here's portions of the synthesis. Let me just pull out this one portion where we're making this episulfide. So what you do is you treat it under these conditions. You get this intermediate, and then you treat it with this double bonded sulfur. You get the episulfide. And then you pull out the sulfur with a thiophile with trimethylphosphite, and that will give you that twisted double bond. And then we go ahead and we finish the route into the nanocar. Lots and lots of synthesis here. This is painful work. You take a graduate student and you chain them to the bench, and you don't let them go until they deliver. You just push and push and push. This is not easy. This is trench work, real trench work. And I'm glad I don't do trench work anymore. I just say, go do it. Here is the actual synthesis that's written in the form for an experienced chemist to make just this one step, this episulfide. From here, this is the hydrazone, to the episulfide. Here's what it looks like. to an oven-dried 3-neck round-bottom flask charged with the hydrazone and magnesium sulfate was added dichloromethane. To this suspension was quickly added manganese oxide at about 5 degrees centigrade. The reaction flask was immediately immersed and stirred in a cold bath ranging from minus 15 to minus 10 degrees C for 1.5 hours. After this period, the reaction mixture was cooled to minus 50 and then transferred to a Schlenk filtration tube connected to an oven-dried 3-neck round-bottom flask. The deep purple filtrate that contained the intermediate 36 was collected and the Schlenk tube was rinsed with pre-cooled dichloromethane, 20 mils at minus 50 degrees. To the flask containing the combined filtrate, thione 35, was added portion-wise until no more nitrogen evolved. The mixture was stirred for an additional half hour at ambient temperature. The mixture was poured into methanol, vigorously stirred in a white precipitate form. The solid was filtered and the filter cake was washed with methanol and dried under vacuum to afford the desired compound. That's a shortened version for people that are experienced. You see the temperature control. You see the solvent control. You can't just willy-nilly switch out solvents. It doesn't work. How do we know? Because we tried many things to get this. Many things. Lots and lots of things. We thought and thought and thought and thought to try what to vary. We didn't just say, maybe we'll just try a little bit of this and a little bit of this. You don't do chemistry that way. Nothing will ever work. But that's what abiogenesis would have to do, because there's no intelligent design agent. How do we characterize it? Well, we look in NMR to try to figure out the bonding patterns. How do you characterize it? What I'm going to show you, you don't have to read. It is the details of what we have to write for the reviewers, for the people in our field, to believe us that we have the right molecular structure. Here's where it starts, part 1. A combination of standard deuterium, proton, C13, and depth 135 C13 experiments in standard 2D, COSY, NOE, HSQC, and HMBC experiments obtained on a 500 megahertz spectrometer yielded the assignment for 38, discussed, the proton NMR, and then we just go through why. And then there's part 2, all right? This is what we have to do. You say, oh, come on, biological systems don't have to do this. Oh, yes, they do. Not on a spectrophotometer, not on a spectrometer, because we buy a spectrometer for a few $1 million and it can characterize many compounds. Nature, the way it does it in a biological system, it has recognition molecules that it is pre-designed so that what you make fits in there. It says, yeah, that's good. It fits. Hand in glove, it fits. If it doesn't fit, it says, bad. And another scrubber molecule, bam, chops that molecule out. That's what happens in nature. But in a prebiotic system, you don't have the scrubber molecule. So all the junky molecules just stay there. And remember, our yields are never 100%, and prebiotic chemistry couldn't have been 100%. Could not have been 100% every time, first reaction. Chemistry doesn't happen that way. Remember, you have to call me out if I'm lying. Have I told a lie yet? Okay. The slow motor to the fast motor. How did we go from the slow motor to the fast motor? Well, we just pulled out a sulfur atom. Let me tell you what a biologist would say. Oh, an enzyme comes in and just yanks out the sulfur, and it just comes in and you get the 5-membered ring. Now, show me the chemistry. How do you do that? There is no known method, period, to do that. Enzymatic, non-enzymatic, no known method. The Ramber-Buckland reaction can't do it when you have a pre-existing double bond there. Can't happen. So what did we have to do? We had to build a whole new segment from the beginning. So here's how we made the slow motor rotor part. Here's how we made the fast motor rotor part. Different chemistry, different starting material, different chemistry to do it. Nature would have to do the same thing. It makes the wrong thing. Uh-oh, this is not going to the single cell that I want. I'm going to have to redo this. Well, remember, it doesn't have a brain. It just has to hope that in some other pool, in some other cave, something else is brewing. It's hard to think of this. This is why chemists go, we have no idea. And then biologists will say, oh, you know, this happens, that formaldehyde is the basis of all carbohydrates. Okay, how? You see the frustration that I have to go through? So here's an 11-point summary of the nanocar syntheses. We had a choice of target. We had to select the target. We had solubility problems like crazy. We had 50 different solvents to choose from. Nature has one, water. It's hard to do organic chemistry in water. You have to build molecules that project hydroxyl and amine groups out. Very difficult to do organic chemistry in water. And poor nature is restricted to that. We had molecular flexibility. We had to build it so that it could climb up. We had to put addends on it to keep it soluble throughout. Nature has the same problems. When we added a motor to the nanocars, the former chassis were not sufficient. We had to redesign the chassis to accept the motor. When we desired to go from the slow motor to the fast motor, though the stator was reusable, the rotor wasn't. The rotor had to be redesigned from step one and then restarted at the beginning. Why? Because we stored the intermediate in the freezer. Where does nature store that thing? Well, that's in a pool over here. It's in another cave over here that you got the rotor. How do you get the two to mix? Remember, abiological world. This is a tough world. Once we changed the wheels to make them suitable to work with the motors, we learned that the new wheels were not compatible with the gold surface that we had been using. So we had to change to a different surface. Most of our reactions are not quantitative. Many yield in the range of 50 to 60%, sometimes lower, and these are our optimized yields, where we tried repeated reactions under different conditions to afford the yields at those levels. The first time reaction yields were always lower or even 0%. We have the convenience of ordering many of the requisite reagents to our starting syntheses. This is JIT, just-in-time procurement. System permits us to have most chemicals at our doorstep in 18 to 24 hours from the day we call it in. Nature doesn't have this. Oh, well, boy, it'd be good to have this. Well, it's a prebiotic world. The companies don't exist. Where are you going to call in the chemicals just in time? It has to be just in time. It has to be there when you start, and your starting material doesn't sit there and wait a few million years. It doesn't. Why doesn't it wait? Because there's something called oxygen around, and that decomposes the organic molecules within a period of weeks or months. You can't wait around for the next pool over to start making something because you all of a sudden need it. Reagent addition order is critical, as seen in the detailed experimental protocols. In other words, A needs to be added before B, and then C, each at its own specific temperature, and with its own specific reagents. The parameters of temperature, pressure, solvent, light or no light, pH, oxygen or no oxygen, moisture or no moisture, has to be carefully controlled to build complex molecular structures. We don't know how to do it any other way. Oh, but an idiotic biological, pre-biological world could do it. The characterization of each step is critical. Once the desired product is synthesized, it can take much longer to properly characterize the product than it did to make it. Now, using nature's constructs, some may contend that I did not use nature's building blocks, such as carbohydrates, amino acids, nucleic acids, and lipids. I concede I took the easy route and used simple synthetic molecules, not nature's far more complex compounds, where chirality and diastereoselectivity can be enormously problematic in synthesis. This is where you have mirror image handedness. I didn't have to deal with that in the nanocars because we only had one stereogenic center, one motor per car. Didn't matter how it landed, whether the motor spun to the left or to the right, didn't matter. It was still going to work on the car because we only intentionally put one motor in there. Thus, here we will now consider nature's building blocks, showing that many of the common parameters hold, yet they become far more difficult for prebiotic systems than for synthetic chemists today. Let us begin at ground zero with the construction of one basic building block of life, carbohydrates. Carbohydrates are the backbones to string together nucleic acids, which are needed for DNA and RNA in the codes of life. So long before you have DNA and RNA, you got to have carbohydrates to string them together. DNA and RNA are like, those are beads hanging on a string. You got to have the string, got to have the carbohydrates. And their polymers serve as recognition sites for cells to communicate with each other and as food sources for living systems. One generally concedes that making a specific polypeptide or enzyme in a repeated fashion requires a polynucleotide blueprint. So we will just consider the basic building blocks, carbohydrates, prior to their polymerization, which requires enzymes. So a choice of target. Here's the 11-point details now with nature's constructs. The same 11 points in the same 11-point order that I showed. Now we're going to look at it in nature's constructs. A choice of target was needed for the nanocars. How do we know what to target? Toward which structures do we optimize to have an adequately functioning system for a task? For a carbohydrate, how do we choose? Take, for example, the pentose sugars, one of the more common carbohydrate sizes, and that used for DNA and RNA backbones. Pentose sugars have 3 stereogenic centers, 1, 2, 3. So 8 possible isomers. There's all the 8 possible isomers. These are substructures, some being enantiomers, which are mirror image related, and others being diastereomers, which involve subtle orientational differences. So these 2 are enantiomers. This to this would be a diastereomer. And all are chiral, meaning that they have non-superimposable mirror images. But what if we do not know the target? Then the complexity of the problem would certainly be compounded. But let's assume we know the target. We want to make the pentose sugar, which we know we need for DNA. So we'll give prebiotic chemistry that. Specifically, we need the 5-carbon sugar, or D- ribose, D-levothyroate ribose in particular, selected from the set of 8 possible pentoses, which I just showed you. Further, for DNA, it has to have 1 hydroxyl group deficient, or deoxyribose. If it is not, then it will be suitable for RNA, but far less stable. But prebiotic systems never knew any of this. There was a blinded pathway to hosts of products, somehow selecting the one desired long before any selection agent could have been biologically available. And what are the selection criteria? It's hard to know if we don't know the target. The prebiotic problem is hard. And even if the target were known, the selector would be another molecule at least as complex as the desired analyte. And what selected the selector? Solubility problems were confronted in the nanocar. Same problems for abiogenesis. Hard to get organic compounds soluble in water. Molecular flexibility, a less rigid chassis was needed since a movable nanovehicle over long ranges was never obtained due to restrictions at step edges. This was part of the redesign needed, where we went back to step 1. Prebiotic chemistry would have to do the same thing, redesigning structures when desired function, and what is desired function since no target is foreseen, was not realized. Thus, much of the work that was done to that point would likely have to be discarded, increasing the difficulty for the prebiotic system. When we added a motor to the nanocars, the former chassis were not sufficient to accommodate the motors. Likewise, in prebiotic chemistry, this again sends the system back to the beginning. It can't just throw in a change. It has to go all the way back to the beginning. You mean all of these millions of years I've been waiting to get to here and you're just going to throw me out? Yep. Go back to the beginning. When we desired to go from a slow motor to the fast motor, though the stator was unusable, the rotor was not. Though the stator was reusable, the rotor was not. The rotor had to be redesigned from step 1 so as to become a fast unidirectional rotator. In a prebiotic system, for small changes, we cannot use a blackboard to delete atoms and reinsert atoms elsewhere. This is what a biologist does. Erase. And how did that happen? Enzyme. Oh, enzyme. Okay. Give me a clue on the chemistry how that enzyme did that. Often redesigns are needed which send the system back to the near origin of the synthesis. This is further exacerbated by the fact that there is no specific target in abiogenesis. Just as our motor no longer functioned when the original wheels were present, and we did not realize it until the synthesis was complete, and we tested it, any prebiotic system is destined, at least some of the time, to experience such disappointment, thereby sending it back to the beginning. But it does not know how to stop its current course of progress, or why to stop. The prebiotic system would continue to make derivatives of non-functional entities, because nobody told it when to stop. So it just keeps going. To get chemical reactions occurring in high yield is difficult. In our synthetic case, we designed reactions to minimize diastereomeric mixtures that can be nearly impossible to separate. Hence, even with our well-developed separation protocols and equipment, we try our best to avoid undesired diastereomers because the separations are too time-consuming and expensive. Plus, they waste a huge amount of starting materials, generating unwanted products. And enantiomeric separations are all the more difficult. Nature has chosen a far harder route, using predominantly one enantiomer or homochiral in a system with multiple stereogenic centers. Here's three compounds: formaldehyde, glycoaldehyde, and glycoaldehyde phosphate. Three very simple organic compounds. Recognizing that carbohydrates are the key building block, and in particular the 5-carbon pentoses, since they form the scaffolding for both DNA and RNA, Eschenmoser. And Eschenmoser is a great synthetic chemist. I have admired Albert Eschenmoser since I was a graduate student. And the man now is in his 90s. He is a superb synthetic organic chemist. I am not coming against his synthetic talent at all. He is excellent. Let me show you what the most excellent chemist can do in a prebiotic chemistry proposal. Eschenmoser has spent years suggesting prebiotic routes to those compounds. The direct synthesis of the pentoses was not successful when starting with a presumed simple natural precursor, glycoaldehyde, and using the foremost reaction, which is the base-catalyzed coupling with formaldehyde in the presence of a divalent cation such as calcium. That means you take formaldehyde, you take calcium, and you take a base, and you get glycoaldehyde. Cleverly, Eschenmoser, being the master synthetic chemist that he is, did not start with formaldehyde, nor Steimer glycoaldehyde, but with the phosphorylated glycoaldehyde to make it a more suitable coupling partner. This is where he started. If you go with the foremost reaction from formaldehyde to glycoaldehyde, that reaction goes to the dimer. It doesn't stop there. And then it'll go to the trimer. It does a polyaldol reaction. Boom, 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 boom. Goes to polymer. How do you stop it right there and then phosphorylate? Not easy to do, but Eschenmoser can do it. Trained synthetic mind. Since glycoaldehyde is the dimeric form of formaldehyde, one would first have to make the dimer of formaldehyde and stop there prior to the further aldol chemistry taking place in the foremost reaction. Nevertheless, a good organic chemist can design conditions to isolate the product, purify it, and then proceed, as did Eschenmoser. Can phosphorylation then occur? Sure, under high concentration of phosphorylating agent, but not in the presence of strong base, such as was required for the formation of the glycoaldehyde itself. So the glycoaldehyde needs strong base, then it needs to find itself away from the strong base. Then it needs to find itself away from the strong base to stop the reaction. While away from the strong base in near neutral solution, it's phosphorylated with 4 molar equivalents of amido triphosphate in near neutral aqueous solution, while further in the presence of magnesium ions. Now, now that the proper phosphorylation is complete, and the glycoaldehyde phosphate is isolated and purified, Eschenmoser then exposed the isolated and purified glycoaldehyde phosphate to 2 normal sodium hydroxide, which is very basic, at a concentration of 0.08 moles per liter. So the starting formaldehyde was in strong base, then the product was isolated, liberated from the strong base, then exposed to a neutral solution of amido triphosphate, which the researchers made separately, then isolated the product, glycoaldehyde phosphate, and conveniently exposed it again to strong base. Even with all its masterful designs, the mixture did not produce the desired pentose, but mostly the racemic hexose triphosphates. Then, using a stroke of superb synthetic insight, Eschenmoser placed the glycoaldehyde phosphate in strong base, then added 0.04 moles per liter of formaldehyde to obtain 40 to 50% of mostly racemic pentose diphosphates. That's the 8 components that I showed you formerly, with 2 phosphates at the C2 and C4 positions. That means that there are 8 possible isomers, 4 diastereomers in that 40 to 50% yield mixture. The desired racemic ribose 2,4-diphosphate, approximately 15% from the total reaction. Also racemic arabinose 2,4-diphosphate, racemic glycose 2,4-diphosphate, and racemic xylose 2,4-diphosphate, where racemic signifies a 1-to-1 mixture of the 2 enantiomers, along with 11 other unidentified carbohydrate species, all bearing their enantiomeric partners, meaning 22 other unidentified species from that 40 to 50%, with the remaining 50 to 60% being unidentified non-volatile compounds such as higher oligomers and polymers. This is a mess, an absolute mess. The best synthetic chemist makes a mess, but in a cave with nobody there. In a prebiotic milieu, the correct isomer would have to be selected to go forward. But selected by what, since this is long before any enzymes could have been biologically tailored? Even with the expertise of the researchers involved, the data more easily argue that any related prebiotic process is unlikely to afford the requisite carbohydrate building blocks. Their results better argue against this being the root, since such careful controls had to be enacted by the masterful synthetic chemists to afford even gross mixtures of the product. In the synthesis of the nanocars, we had convenient just-in-time delivery of chemicals and storage of our intermediates in safe, stable conditions away from light and air in freezers. Using the routes described above to the pentose compounds, Eschenmoser candidly explains that, quote, that the preferential formation of the desired ribose 2,4-diphosphate under the reaction conditions employed is a result of kinetic reaction control. Uh-oh. What that means is that extended time is working against the researchers. In the laboratory, as anywhere else, it is essential to stop a reaction before the desired product degrades. The beautiful stuff that he got in just a trace amount of yield with all us decomposes over time. Oh, but time is our friend in building a carbohydrate that's eventually going to be used for DNA and RNA. Time is our friend. No, time is your enemy when you're making kinetic products. Thus, the desired compound was not the most stable of the isomers, and even with the cleverly designed experiment that started with the proper glycoaldehyde rather than the more rudimentary formaldehyde, that was further intelligently and conveniently phosphorylated to the glycoaldehyde phosphate, the desired racemic ribose 2,4-diphosphate degraded under the very controlled conditions under which it formed. Furthermore, while Eschenmoser teaches that, quote, the total amount of the 4 diastereomers is largely unchanged, unquote, that's not entirely correct. I went back to his data and read the experimentals. His own data shows that the combined yield of the 4 diastereomers dropped from 34 to 30% over the course of an additional 22 weeks, hence a relative 12% loss of the pentoses over 22 weeks. Thus, after a few years, which is a brief moment in time by prebiotic terms, there would be little, if any, pentoses left, let alone the more rapid loss of the desired ribose 2,4-diphosphate. Where does the material go? Likely to extended oligomers and polymers, which is common in organic reactions and especially common in extended aldol-based processes, proceeding to the point of being insoluble in solution or generating a viscous mixture of ill-defined polyols. Junk. It's called junk. Crap. This is what any chemist will call it in a laboratory. What'd you get? Junk. I know what that is. It's a bunch of polymeric stuff. After all this work on the pentoses, which mimic those seen in nature for DNA and RNA, Eschenmoser switched to what he called homo-DNA, the oligonucleotide composed of the 2,3-di-deoxyallose-pyranose, a 6-member ring instead of DNA's 2-deoxyribose-furanose, 5-member ring for the carbohydrate backbone. That gave him added stability he needed for subsequent base pairing studies. Even with as good as he was, he couldn't make it. So he made a different one, a 6-membered ring, whereas we know biology uses 5-member rings. He couldn't figure out how to do it. And he humbly conceded that, quote, from an etiological perspective, the work on homo-DNA was regarded as a model study only, albeit one that proved to greatly facilitate our entry into base pairing prognoses, the development of methodology for oligonucleotide preparation, and the interpretation of base pairing characteristics of alternative nucleic acids. Prebiotic chemistry is extremely difficult to perform even with the world's best synthetic chemists like Eschenmoser, so he chose a more convenient model to study. Reagent order of addition is critical, as we saw reading Eschenmoser's experiments, and is seen in detailed protocols. A needs to be added before B, and then C, each at its own specific temperature. The parameters of temperature, pressure, solvent, light or no light, pH, oxygen or no oxygen, moisture, no moisture, has to be carefully controlled to build complex molecular structures. That's even before you assemble them into a complex nanosystem. Unless one can devise sophisticated promoters or catalysts that are stable in air and moisture and can work at common atmospheric conditions, precise control must be maintained. The characterization at each step is essential, and even more so if we ever have to bring up more material synthesis from the rear. What do? You work on a synthesis and you run out of starting materials and you go back to your notebooks and you say, okay, well, this is how I made it, and you make more. And you pull it up from the rear and you then do a few more steps. You run out of starting materials, you go back. Right? Synthetic chemists, isn't that what you do? Every day you got to do that, bringing up more starting material from the rear. Tell that to prebiotic chemistry. Oh, you just ran out of starting material, make more. I never kept a lab book. Prebiotic chemistry doesn't keep lab books. I don't know how I got here. I just got here. You're going to have to figure it out all over again. Ab initio. Therefore, small changes in ultimate function demand major rerouting in synthetic approaches. All changes when doing chemistry are hard and cannot be done through hand-waving arguments or simple erasures on a board. Laborious and intentional elements of forethought are required. All right, let's say, okay, well, you're only considering carbohydrate sugars. Okay, let's look at more than just sugars. You want to go to another compound? And this is going to be shorter, so bear with me. If you leave, God bless you. I understand. I would leave too. More sophisticated proposals to prebiotic feedstocks have arisen since the often quoted work of Eschenmoser. It is generally proposed that there must have been a chemical means to generate an information-bearing molecule, such as DNA or RNA. Since the 1960s, many have suggested that the polymer is RNA rather than DNA, leading to the RNA world hypothesis that was really promoted and pushed in really beautiful work in the 1980s. Aside from the fact that the RNA backbone is very unstable relative to the DNA, chemically activated ribonucleotides, through careful manipulation of the structure, can polymerize to form RNA, but of course with no prescribed sequence. And as observed by Eschenmoser and many others, the coupling of ribose with the nucleotide is the first step, and even those engrossed in prebiotic research have difficulty envisioning that process, especially for purines and pyrimidines. Sutherland and co-workers, and again, Sutherland is an excellent, we're talking about the best organic chemists out there. Sutherland and co-workers have insightfully proposed that pyrimidine ribonucleotides can form short sequences using arabinose amino oxazoline and then hydronucleosides intermediates, all from simple compounds such as cyanamide, cyanoacetylene, glycoaldehyde, glyceraldehyde, and inorganic phosphate. The use of inorganic phosphate changes the previous proposed conditions to a pH-buffered solution, thereby slowing decomposition pathways. But the work itself shows the intricacies required to afford the desired reaction products. The conditions are so cleverly selected that the researchers candidly concede that, although the issues of temporarily separated supplies of glycoaldehyde, that's just-in-time scenario one. Remember, abiogenesis never had this, but he's inserting it. And glycoaldehyde, that's just-in-time delivery scenario two, remains a problem. A number of situations could have arisen. Prebiotically, what and where? That could result in the conditions of heating, his carefully controlled step one, heating up to 60 degrees centigrade, and progressive dehydration, carefully controlled step two by lyophilization, which is water removal by ice sublimation under reduced pressure at 0.001 atmospheres. Find that in nature. Rehydration, all of a sudden, water appears. We took away all the water, now water appears. Rehydration, carefully controlled step four with a precise adjustment of pH. And ultraviolet irradiation, careful control step five with selected 254 nanometer light. How are you going to get just 254 nanometer? Further, there were also multiple purification steps, careful control step 6, and ion exchanges using commercial resins, careful control step 7. All of this is for the synthesis of one set of a mixture of adducts, all in racemic form. The forethought required to afford even a mixed set of selected structures en route to the array of molecules needed to form randomly sequenced, that means non-information bearing, RNA, is painfully improbable. When I say non-information, this is what I mean. I give you the alphabet, the 26 letters on one side. On the other side, I have the book, War and Peace. Any difference between these two? You'd have to shake around those 26 letters a lot of times to get War and Peace. War and Peace is information encoded. Everything they've made is just one part of an alphabet of many letters and with no information. Then in 2015, Sutherland, we're talking Nature series journals. This is being published in the best journals of chemical science. In 2015, Sutherland and co-workers pointed out that, quote, a minimal cell can be thought of as comprising informational, compartment-forming, and metabolic subsystems. Okay, I'll give them that. That's a very, very, very simplified view of a cell, but we'll give them that. They also astutely acknowledge that to date, prebiotic chemistry has made ambitious extrapolations. Wow, okay. To imagine the abiotic assembly of such an overall cellular system or subsystem, however, places great demands on hypothetical prebiotic chemistry. So there, Sutherland says, you know, we've really had a lot of conjecture here. But then, yet this revealing comment by Sutherland and co-workers is coupled to the disclosure of a new experimental finding showing that, quote, the precursors of ribonucleotides, amino acids, and lipids can be derived by the reductive homologation of hydrogen cyanide and some of its derivatives. The key reaction steps are driven by ultraviolet light using hydrogen sulfide as the reductant and can be accelerated by copper-1, copper-2 photoredox cycle. Albeit superb synthetic insight and exquisite experimental work, the bold extrapolation associated with this new finding that all cellular subsystems could have arisen simultaneously through common chemistry has raised the level of suppositions from mere molecule types to now complex subsystems where molecules are working in concert toward a common functional goal. What I mean by this is Sutherland has now said, yeah, we've had a lot of real conjecture here. But one could think of a way to have all this assembled directly into the nanocar. That's what he has said here. And this is published in Nature series journals. This is a remarkably higher level extrapolation that pains not only the synthetic chemists, but those involved in nanosystems and molecular device research. Compositions of a few molecule types, or even all of them, do not constitute a cellular subsystem. It is essential to emphasize that the authors only prepared precursors to the ribonucleotides, amino acids, lipids, not the actual molecules. So the gross extrapolation is unacceptable, and its gross extrapolation and its acceptance into the best chemical journals is all the more disconcerting. When reading the precise protocols for Sutherland's suggested prebiotic-like precursors, one captures high-level sophistication, expert synthesis prowess, and remarkable ingenuity of the researchers. Some reactions were conveniently run at room temperature, others at 60, others at 100 degrees, then washed with ice-cold water. The molecules prepared by these supposed prebiotic routes were not used, but they had to be made more cleanly and in larger scale using purely synthetic methods of organic solvents, such as and organic reagents and solvents, such as Lawson's reagent and tetrahydrofuran, to quote, to simplify the handling procedures, unquote. No, it's because you couldn't get it any other way. Just-in-time and precise order of addition protocols were used over and over again. One sees precise pH adjustments through the syntheses, use of ion exchange resins, and separations from the reaction mixtures because proceeding without separations would have destroyed the carefully prepared products. The preparation of cyanoacetylene on copper-1 was suggested as a way to conveniently prepare and store it for use when needed. Copper chloride was mixed with potassium chloride to generate the Newland catalyst at 70 degrees centigrade. Then a separately generated source of acetylene gas was prepared from calcium carbide and water. The gas was bubbled into the Newland catalyst to prepare acrylonitrile. Acrylonitrile is a very unstable molecule. And you have to work with it carefully to inhibit its polymerization, which was then treated with potassium cyanide for 1 hour. Then 5 equivalents of ammonia at 13 molar ammonium solution adjusted to pH 9.2 with sodium hydroxide to generate the desired aminopropionitrile. Try to do that in a cave somewhere. This is just a sampling of the preparations that are difficult even for the most skilled synthetic chemists, preparing very simple precursors to a few of the many molecules and building blast types. All of them bore no clean stereoisomerism. All right, now let's just change directions. Let's just give prebiotic chemistry proposals a chance. You say, Tour, you're just too hard on them. Okay, let's assume that all the building blocks of life, not just the precursors, but all the building blocks, could be made in high degrees of purity. including homochirality, where applicable, for all the carbohydrates, all the amino acids, all the nucleic acids, and all the lipids. Let's further assume that they're comfortably stored in cool caves, away from the sunlight, away from oxygen, so as to be stable against environmental degradation. Let's further assume that all existed in one corner of the Earth, not separated by thousands of kilometers, and not on different planets. All in the same cave. We'll give them that. All in the same cave. All chiral and pure. We'll give it to them. And that they all existed not just in the same square kilometer, but in neighboring pools where they can conveniently and somehow selectively mix as needed. Now what? How do they assemble? Without enzymes, the mechanisms do not exist for their assembly. It will not happen, and there is no synthetic chemist that would claim differently, because to do so would take enormous stretches of conjecturing beyond all that is realized in the field of chemical sciences. You can't do this in chemistry. All right, now let's assemble the dream team. You got good professors here? Let's assemble the dream team. Let's further assume that the world's top 100 synthetic chemists, top 100 biochemists, and top 100 evolutionary biologists combine forces into the limitlessly funded dream team. The dream team has all the carbohydrates, lipids, amino acids, and nucleic acids stored in freezers in their laboratories. Taking it out of caves, you can use their labs. All are in 100% enantiomeric purity. Even give the team all the reagents they wish, the most advanced laboratories, and the analytical facilities, and complete scientific literature, and synthetic and natural yet non-living coupling agents. Now ask the dream team to assemble their building blocks into a living system. Nothing complex, just a single cell. They'll merely scratch their heads and walk away frustrated. Remember, call me a liar if I'm lying. So let's help the dream team out by providing the polymerized forms, the polypeptides, all the enzymes they desire, the polysaccharides, all the carbohydrates, all hooked up. DNA, RNA, in any sequence. I'll even give you the sequence, whatever sequence you want. You tell me the sequence, you got it. Ask the dream team to assemble those into a living system. The level of sophistication in even the simplest of possible living cells is so chemically complex that we are even more clueless now than with anything discussed regarding prebiotic chemistry or macroevolution. The dream team will not know where to start. Moving all this off Earth does not solve the problem because our physical laws are universal. You see the problem for the chemist? Welcome to my world. This is what I'm confronted with every day. And then people will flippantly say, oh, these are made from small organic compounds by the foremost reaction from formaldehyde. Oh, okay, I get it now. So here's a summary. Those that think that scientists understand the details of life's origin are wholly uninformed. Nobody understands. Maybe one day we will, but that day is far from today. So to make ad hominem attacks upon those who are skeptical of the science to date can be inhibitory to the progress of science. Would it not be helpful to express to students the massive gaps in our understanding so that they, as the next generation of academic soldiers, could seek to propel the field upon a firmer and possibly a radically different scientific basis, rather than upon increasingly ambitious extrapolations that are entirely acceptable and unacceptable in the practice of chemistry? The basis upon which we as scientists are relying is so shaky that it would be best to openly state the situation for what it is, a mystery. That might catalyze some fresh scientific thought on abiogenesis. I saw a presentation by a Nobel Prize winner modeling the action of enzymes. And I walked up to him afterward. I said, I'm writing an article entitled Abiogenesis Nightmare. Where do these enzymes come from? He says, these things are synthesized biologically. I said, no, starting from the beginning, where did these things come from? He says, what did you write in your article? I said, it's a mystery. He said, that's exactly what it is. It's a mystery. It's a mystery. Why do so few chemists speak up regarding difficulties with origin of life? Few think about it intently. They're just too busy with other things. I mean, we got one synthetic chemist here, the guy's trying to get tenure. I mean, he's got a lot of things to think about. He doesn't think about abiogenesis. How often do you wake up in the morning thinking about abiogenesis? Not very often. Yeah. You got a lot of other things to do. You got a lot of emails to answer. They think that someone else understands. That's what they think. They think someone else understands. It's just like you. You think someone else understands. I'm telling you, nobody understands it. It's viewed as the only ball game in town. Okay, we don't have another thing. You shouldn't shoot it down, Tour. You shouldn't shoot it down until you have an alternative proposal. Oh, really? Is that what we teach our students? Do we teach our students that you can't contest with that mechanism until you have a better one? No, if the mechanism doesn't explain the facts, you discard that mechanism or you change it to fit the facts. That's your proposed mechanism. You don't say until you have something better. No, you just shoot the thing down. And also for fear of being ostracized and fear of reprisals. Some think it's even justified to go after people who won't buy into this. They think it's even justified. They deserve to be disenfranchised. It's a sad state of affairs. When will the scientific community confess to the world that they are clueless on life's origin, that the emperor has no clothes? When will they say this? Here's what Max Planck had to say. We have Planck's constant, the great physicist. Here's what he had to say. A new scientific truth does not triumph by convincing its opponents and making them see the light, but rather because its opponents eventually die. This is what I think is going to happen. I don't think that those that are entrenched and keep saying that there's plenty of understanding when there's not, and when you push them on it, you see not only that there is little, that there is none. None. You're going to have to wait for them to die. Here's the hope that I see, though. Science is self-correcting. If abiogenesis proposals are correct, the chemical descriptions will become evident. As of today, in my opinion, there is only evidence on how it did not occur. So further investigation is warranted. We must continue to do research in this area. And here's the great hope. The younger generation has a deeper sense of social fairness and justice, and they're less impressed with conformal academic fluff. Once the young progress into positions of influence, they will correct the injustices that have been thrust upon those that dare to question the mainstream scientific establishment. If you dare question the mainstream scientific establishment as I am doing today, you will be held out of certain societies. How do I know? Because I was told I wouldn't get into certain societies because of my views on these things. I was told this to my face behind closed doors. And I said, then I will speak up. Then I will speak up. And the words of Groucho Marx are greatly comforting to me. When he said, I don't want to be part of any organization that would have me. You young people have a much greater sense of social justice and fairness than my generation has. You feel the pain when people are disenfranchised. You're going to get into positions of influence. Change it. Change it. Say it when you don't understand it. Say it, I don't understand it either. And let there be a groundswell of correction here. That's the end of The Origin of Life, an inside story. Thank you.
Thank you very much for that. My name is Derek. Thank you for a very informative and interesting presentation. My question is very brief. It is, what would you say to someone who suggested that yours is a God of the gaps of our understanding, or the things that we don't know? That is where you would find answers to the questions that you don't have results for.
So I didn't bring up God in this at all, I think. So that's not a part of this talk. So, you know, if you want to inject God, you can do just about anything. You could speak it into existence if you wanted to inject God. But I've intentionally taken this talk and I've not inserted God. And to say, here is the scientific problem that we're up against. It is clearly a very, very difficult problem. The origin of life is the most difficult question, far more difficult than Darwinian evolution. The origin of life is a very, very hard problem. Something that you have to solve before you can even discuss evolution. You have to solve this one. If you want to inject God, God can speak it into existence, but I've not done that. Okay?
Thank you very much.
You're welcome.
So actually, what I want to really talk about is actually yesterday I was checking about that. It's from your website. You have an article called "Evolution versus Creation," and you mentioned that you don't want to be labeled as an intelligent designer proponent. And you also said as, of course, I know that microevolution it indeed exists, but what your point of view on that? Is anything you change? Because what I see from these articles, a lot of the resources here is quite updated, is back in 2000. So I don't know.
That I was just trying to give the background, because sometimes if you say macroevolution versus microevolution, they say, don't try to separate these terms. But what I was showing there is I didn't generate these terms. These terms came out of the mainline literature. That's it. I'm not separating the terms. That's all I was saying there. I wasn't using that as a proof text. I was only saying that the literature brought that forth. And that article's been up there for 10 years.
That's
my article's been up there for 10 years.
Yeah, so what's your opinion for now, since it has been...
It hasn't changed. It's exactly what's written there on the web is exactly where I stand now.
Okay,
hasn't changed. So if you've read that article, you know exactly where I stand.
Okay.
You've got it in your hand. That's exactly where I stand.
Okay.
No, I'm just telling you, I haven't changed. I haven't evolved. I'm right here. Okay?
Okay, thanks.
Thank you.
Hello, sir. Hello again.
Hello again.
Yeah. My name is Sunny. I'm a first-year computer science student at Waterloo. Yeah, thanks for coming.
Did I get the computer science right?
Yeah.
Were there things that I talked about coding?
I'm a first-year student.
Okay.
Yeah. I actually have a very strong connection with Rice University because they rejected me last year. But
our loss.
Yeah, but thanks for coming. Anyways, my question is going to be a lot less sophisticated than the previous ones. I just want to ask, because you talked about that you have an opportunity to talk to a lot of chemists who also disagree, who also think it's very hard to do the stuff you talked about.
Yes.
So do you know if any of those people speak out about ideas like this, like you?
Absolutely not. They will never speak up about this. In fact, when they share with me, they've even said things, please don't quote me. And I say, I will never quote you. You know, I quoted them, but I didn't tell you who it is. Because they fear what will happen to me happens to them. Look, if they shoot at me, I've been very blessed to have done a lot of things that have impacted the chemical literature. If they do this to me, to exclude me from societies for this position, what do you think they would do to an assistant professor? And I don't blame them for standing back. I don't blame them for not wanting to talk about these things. But even the big shots, even the people in the National Academy, they don't want to be quoted because they don't want to have to go through what I'm going through by saying the emperor has no clothes. You'd think that the scientific community would be more objective and they wouldn't do this to each other. Au contraire. They really do this to people. And in fact, it's worse than just doing it. They will even justify that we should do it to you. It's terrible. It's terrible. It's not that they do this quietly. They will do it and even tell you that the disenfranchisement is justified. That's how bad you are. Bad. It's a shame that science is this way. You're going to correct it. You're going to correct it. Your generation will correct it because your view of social justice is much finer than my view. And it's not the young guys that are doing this. It's the old guys that are doing this. Okay?
Thank you.
All right.
So thank you for giving what I think is the most technical Pascal lecture I have ever heard. You do make a very interesting case that the origin of life is an exceptionally difficult and unsolved problem. Do you think there are some scientific problems that are just too hard for us to ever solve?
No,
Because?
because I am amazed at human ingenuity. What Einstein predicted, and now we have just seen in the rippling of the gravitational fields, is extraordinary. I am just in awe of this. This may be one of the greatest discoveries in our lifetime. I mean, it's that big. It is.
I know.
Really...
I work in that area. I
Okay.
agree with that.
You agree with me? Okay. I don't work in that area, but I can recognize it.
Yeah.
So, I don't know how much longer humans have to exist, but we've come so far so quickly, and what happens, I talked about our informational rates are doubling every 2 years. I mean, this is phenomenal rates, and the ways that we are progressing. So if we can figure how to keep ourselves from killing each other in wars and nuking each other to death, then we're going to do okay. I think there's a lot of things we can figure out. But while you and I are thinking about this thing tonight, there's people around the world that are thinking how they can cut our heads off. That's what they're thinking about. Mm-hmm. So unless we can somehow mitigate the damage of that, we're going to have a lot of distractions. It's going to make it hard for us to think about these sort of things. But are there problems that are going to be too hard for us to ever fathom? Even the Big Bang. I mean, you work in these areas. 40 years ago, I mean, nobody accepted this sort of thing. And now it's clear. We've got this expanding universe. There's just so much more information that we can gather now. So I think we may get an answer to this at some point. We're nowhere close, but 100 years, 200 years, 300 years, that's a long time in the scientific framework of things. We've come a long way in 300 years in synthetic chemistry. So maybe we'll find an answer. So I'm not going to say that it's going to be too hard to find.
Okay,
I can't say that. How do I know?
all right. So, but I would infer from your statement that you expect one day there is a scientific answer to the question raised up there.
If human beings can survive long enough, yes.
Okay, thank you.
Yeah.
Hi. I was just wondering, how would you incorporate the idea of scale in that, that why isn't it just the origin of life be a matter of probability, considering the massiveness of the universe and the billions and billions of years?
Yes, so the probabilities are so dauntingly impossible. When you talk to mathematicians, the numbers are like 1 in 10 to the 90th. I mean, it's huge, huge improbabilities. Now, this you have to do many, many, many times over and over and over again, and it is such a cop-out to say, well, you got 14 billion years. The Earth is, 5 billion years or something. I mean, 5 billion years, a lot can happen. I mean, this is such a cop-out. These are the problems you got to face. The probabilities are so daunting against you. And then what I'm saying is, even if you got it all, that's why I assembled the dream team, even if they had it all, then what? We're clueless. We're absolutely clueless. And I'm telling you, you could wait billions of years. It's not going to happen. Not going to just happen by anything that we know now. So if you want to chalk it up and just say it's a probability, many mathematicians would disagree with you who've looked at just sheer probabilities. But what I'm showing you is the difficulty of this, the utter difficulty. And I didn't even talk about all the decomposition that occurs. So you get to a certain point, it sits out for 10 years, which is a nanosecond in geological time frames. In 10 years, an organic compound exposed to the air is gone. It is history. So everything you work to that point is just gone. So it's billions of years of stuff working against you is what it is.
All right, thank you.
Okay.
I'd like to thank everybody for coming out tonight and last night as well. Hopefully you enjoyed yourself. And I'd like to also thank once again our speaker, Professor Tour. Please join me in thanking him once again for his talk. Thank you.